In the process of respiration in plants and understanding the process, we have studied about the respiration, for what use it is, that is the cellular respiration and all, and also the anaerobic breakdown of the food material or the glucose. Then, moving a step further, in this video, we are going to study the aerobic oxidation of the pyruvic acid. The pyruvic acid, which is formed as a result of the glycolysis process. Glycolysis process is common for both aerobic and anaerobic respiration. That is the first step. After that, the pyruvic acid is formed due to glycolysis, and the fate of the pyruvic acid, that is the further breakdown of the pyruvic acid, is dependent upon whether the oxygen is available or it is in absence of oxygen, or whether it is present, whether the oxidation takes place in presence of oxygen. So we are going to study today the breakdown or the aerobic oxidation of the pyruvic acid in this video. Now, under the aerobic condition, the pyruvic acid which is formed during glycolysis enters the cycle which is known as Krebs cycle. So, we talk about the fate of the Krebs cycle, talks about the oxidative breakdown, or it is the oxidation of pyruvic acid in aerobic condition. Oxidation but aerobic condition. So the Krebs cycle talks about the breakdown. But before the pyruvic acid enters it enters into the Krebs cycle, it moves from the cytosol or the cytoplasm to the mitochondrial matrix. So, we know the pyruvic acid which is formed in the cytoplasm, this is formed in the cytoplasm, it enters into the mitochondrial matrix. This is the outer lining of the mitochondria. This is the infoldings are the inner. So the pyruvic acid moves from here and it forms acetyl coenzyme A. That is the first step. We'll be studying about it. And this acetyl coenzyme A then enters into the Krebs cycle. So the process of Krebs cycle actually takes place within the mitochondrial matrix. Whereas the synthesis of the ATP occurs in the inner membrane of the mitochondria. So this is the inner membrane of the mitochondria. ATP synthesis will take place over here, but the Krebs cycle will take place within the matrix of the mitochondria. Since ATP is generated in the mitochondria, that's why mitochondria is very commonly called as the powerhouse of the cell. Now, let us study about the various steps which are involved in the formation of or the release of ATP from the mitochondria. The steps involved are that first step when we talk, the pyruvate or the pyruvic acid which is formed by the glycolysis enters into the mitochondrial matrix. So here the pyruvate enters into mitochondrial matrix. Here, after entering into the mitochondrial matrix, it undergoes oxidative decarboxylation through a series of complex reactions. So the pyruvic acid enters into the mitochondrial matrix, and here it will undergo several reactions, that is oxidative decarboxylation. Now, when we talk of decarboxylation, oxidative decarboxylation, it is these are the oxidation reactions in which the carboxylate group is removed and carbon dioxide is formed. So the using oxygen. So oxidative decarboxylation you can make out from the name, that is the removal, decarboxylation, that is the removal of the carboxylate group, and there is a formation of carbon dioxide using the oxygen. So it is a series of complex reactions, and the reaction take place in presence of enzyme pyruvic dehydrogenase. Now, this step from the pyruvic acid to the formation of acetyl coenzyme A using pyruvate dehydrogenase is the most important reaction, and there are 5 essential cofactors for this reaction. The five cofactors are thymine pyrophosphate. So the first cofactor when we talk is thymine pyrophosphate, magnesium, NAD+, and coenzyme A. Also, the lipoic acid which is required for this particular reaction. So when we talk of, let's talk of the cofactors which are involved over here. The cofactors which are involved over here, first is thymine pyrophosphate. The second cofactor is NAD+. Third is magnesium ion. Fourth is coenzyme A. And the fifth one is lipoic acid. These are the cofactors which are involved and to form the acetyl coenzyme A, and this step is also known as the gateway step, because until and unless the pyruvic acid gets converted into acetyl coenzyme A, the Krebs cycle is not possible. So if we talk of the overall reactions over here, overall reaction of the conversion of the pyruvic acid into the acetyl coenzyme A, let us study about that. Using these cofactors, the pyruvic acid which is produced during glycolysis reacts with in presence of coenzyme A, that is over here, plus NAD+. The NAD ion that helps in the removal of hydrogen and electrons from the pyruvic acid. So this helps in actually removing the 2 hydrogen atoms and also the 2 electrons from the pyruvic acid. In presence of magnesium ions and the enzyme pyruvate dehydrogenase, forms acetyl coenzyme A, that is required, plus carbon dioxide is formed, NADH, and hydrogen is released, hydrogen ion is released. This is the first step or the gateway step in which the acetyl coenzyme A is formed. From here, it will now enter into the Krebs cycle. So this process, that is the pyruvic acid formation, has taken place in the cytoplasm of the cell, that is we call it as cytosol. Due to glycolysis, 2 molecules of pyruvic acid are formed. And here we'll be taking one molecule, studying one breakdown of one molecule of pyruvic acid. using coenzyme A, which is a cofactor NAD+, in presence of pyruvic dehydrogenase or pyruvate dehydrogenase and magnesium ions, the pyruvic acid breaks down into acetyl coenzyme A with the formation of carbon dioxide and NADH, that is reduced again, and hydrogen ion is formed. The product, that is the acetyl coenzyme A, now enters into the Krebs cycle. Now, over here, if we cut short or if we try to understand that way, one molecule of glucose breaks down into two molecules of pyruvic acid. This is 6 carbon, this is 3 carbon. 3 carbon. And this forms 2 NADH and acetyl coenzyme A. This acetyl coenzyme A now enters into the Krebs cycle. This cycle is also called as the citric acid cycle. So we also call the Krebs cycle as citric acid cycle because of the formation of citric acid in the first step of the cycle. Because this acetyl coenzyme A will form citric acid, which is the first product. Also, another name for the Krebs cycle is TCA cycle or tricarboxylic acid cycle. TCA in short we are calling, and it is called tricarboxylic acid cycle because many intermediate compounds are formed in the cycle having 3 carboxyl group. Also, it is important for you to understand that the Krebs cycle or the citric acid cycle or the TCA cycle occurs in presence of oxygen, and the site of the cycle is mitochondria. When we study the cycle actually takes place in 9 steps. And each step involves various enzymes. The first action or the Krebs cycle involves the condensation of acetyl coenzyme A to form the citric acid, and it condenses using the oxaloacetic acid. So the acetyl coenzyme A, which is formed in the first, that is using the cofactors, with the help of oxaloacetic acid gets converted or forms the citric acid in presence of citrate synthase. Now this citrate synthase is an enzyme, this enzyme guides the entire reaction, and the coenzyme A, which was the cofactor, it is removed during the formation of the citric acid. The next step is the dehydration. That is the first step, formation of citric acid. The second step is the formation of cis-aconitic acid from citric acid, and the process is known as dehydration. So during the dehydration, there is removal of the water molecule and from the citric acid, and there is an enzyme which is known as aconitase which guides the reaction. So in the cycle you can see the citric acid, there is removal of one oxygen molecule resulting into the formation of cis-aconitic acid, and it happens in the presence of enzyme aconitase. The second step is the hydration of cis-aconitic acid, and as a result over there we have the formation of isocitric acid from the citric acid. So over there the cycle has been marked as aconitase, showing the removal of the the water molecule. Instead, it has been wrongly put. We will put it as addition, that is hydration of the molecule. So here it is the hydration of the molecule, and this is the first hydration where water molecule is added, and it also takes place in presence of enzyme aconitase, due to which the isocitric acid is formed. Both are the isomeric forms of the citric acid. Now here the isocitric acid, after the hydration, that is the formation of from the cis aconitic acid, isocitric acid is formed. The isocitric acid undergoes the dehydrogenation, and here it is the dehydrogenation one, we have marked as one step and two step. The dehydrogenation one is the first step in which the oxidation takes place, and here two hydrogen ions are removed from the isocitric acid. The isocitric is formed due to the hydration, but from this isocitric acid there is removal of the two hydrogen ions, or we also call them as protons, and the in presence of enzyme over here, oxalosuccinic acid is formed. The intermediate step I have skipped over there. Isocitric acid results into the formation of oxalosuccinic acid in presence of enzyme isocitric acid dehydrogenase, and it is a very small step, because the final product is alpha ketoglutaric acid over there. So the oxalosuccinic acid immediately undergoes decarboxylation, that is the removal of the carbon dioxide. So you can see over there, and it results into the formation of alpha ketoglutaric acid. Dehydrogenation 2 and decarboxylation 2, because one decarboxylation has taken place due to the oxalosuccinic acid getting converted into alpha ketoglutaric acid. So here first removal of carbon dioxide has taken place, and alpha ketoglutaric acid, when changes into succinic acid, then it is again undergoing dehydrogenation and decarboxylation. So here the succinyl coenzyme A is formed, that is in presence of enzyme succinic thiokinase and alpha ketoglutaric dehydrogenase complex. This succinyl coenzyme A gets converted into succinic acid due to the entire process. So it is divided into processes, steps, but we have completely marked as alpha ketoglutaric acid changing into succinic acid due to dehydrogenation and decarboxylation. Over there, the dehydrogenation third is taking place. That means the change of succinic acid into fumaric acid. This is the third oxidation step, that is dehydrogenation 3, where two hydrogen ions and two electrons are removed from the succinic acid. So here there is removal of two hydrogen ions and two electrons over here are removed. As a result, fumaric acid is formed. Now over here, this reaction is guided by an enzyme succinic dehydrogenase. The enzyme is succinic dehydrogenase, and also the hydrogen ions which are released by the succinic acid are used to reduce FAD. FAD is flavin adenine dinucleotide. Flavin adenine dinucleotide will be further used in electron transport, and it is the energy for it will be utilized as NAD. So flavin adenine dinucleotide. In the next step, that is the 8th step, the fumaric acid, due to hydration, Over here, the first hydration has taken place in the third step. Now, eighth step, the hydration of the fumaric acid will take place, and as a result, the malic acid is formed. And this reaction is guided by the enzyme fumarase. After the formation of the malic acid, the dehydrogenation fourth step is taking place. That is again the formation of oxaloacetic acid from the malic acid in presence of NAD plus and malic dehydrogenase. Here the NAD plus is reduced to regenerate the oxaloacetic acid, which was again utilized by acetyl coenzyme A to form the citric acid. So here the complex oxidation of one molecule of pyruvic acid, 3 molecules of carbon dioxide are released. And in the fourth oxidation step of the Krebs cycle, 4 pairs of hydrogen ions and electrons are removed and 3 are utilized in reducing NAD plus and 1 is utilized in the reducing FAD. The NADH and FADH2, the NADH which are produced and FADH2 which are being produced over here are the reducing powers that will be later on utilized to produce the ATP. So here the cycle, why we are calling it as a cycle, because the process starts by the breakdown of acetyl coenzyme A and the formation of FAD, FADH2 and the NADH, and this FADH2 flavin adenine that is dinucleotide will be utilized to form the ATP through the electron transport chain. So there all the production of the ATP in a nutshell is brought about by a series of oxidation reaction and oxidation reduction reactions, and here NADH and FADH2 which are produced during the Krebs cycle will be utilized by an electron transport system to produce the ATP. And this ATP production takes place in the inner membrane of the mitochondria. So the importance of the Krebs cycle is it provides the hydrogen atoms which actually generates the reducing power, and also it also manufactures other fatty acids, amino acids, carotenoids, etc. which are utilized for metabolism. Krebs cycle is very important because it was found by found by scientist Hans Krebs, and is also therefore is named after